Welcome again, everyone, to PXE International's webinar, The Presentation and Progression of PXE in Children. I'm Christine Vokey, Director of Education and Information for PXE International. Once again, you can ask questions by using typing your question into the question box that is on the control panel on the right side of your screen. Technical questions we'll try to deal with if we can, and any other questions that relate to the presentation, I will ask the presenter at the end of the presentation. We are delighted tonight to have with us Emilie Lamb, who will be presenting the presentation and progression of PXE in children. Emilie Lamb works for PXE International. She joined us in August 2007, and she is now the research coordinator for PXE International. Emilie received her Bachelor's of Science in Biology and Chemistry at Wake Forest in North Carolina, and recently she completed her Master's of Science in Public Health, Microbiology, and Emerging Infectious Diseases at George Washington University School of Public Health here in Washington, D.C. Before she came to work with us at PXE International, Emilie worked as a technician in genetics and molecular biology labs. She worked for the NIH and other academic institutions. Because of her background in biology and genomics, she's familiar with the genetics of PXE and hopes to lend her knowledge to continuing research into treatment. Emilie now lives with her husband in Durham, North Carolina. Emilie?
Hello, everyone. I appreciate everyone attending this evening. Thank you, Chris, for giving me that introduction. I did recently complete my master's in public health, and as part of my master's thesis, I worked on this progression study in children affected with PXE. And I do want to mention that I am not a physician, and I really am looking at the epidemiology of children with PXE. And so, unfortunately, I'm not going to be able to answer any questions about symptoms or rates of progression of children with PXE. If you do have questions such as those, feel free to enter them into your question box, and we can have our medical director get back to you about those questions. In this presentation, I'll only be able to answer questions about this particular study that we did on the progression of PXE in children. But like I said, feel free to send questions to us if you need to. And there'll be information about where to send those questions to at the end of the presentation. Okay, so let's go ahead and get started. So generally, the onset of PXE signs is that early signs are usually noticed on average between the ages of 15 and 25, and these signs usually include bumps or papules on the neck, and then changes to the retina, such as peau d'orange or angioid streaks. However, we've seen in most of our studies that diagnosis is usually delayed by about 10 to 12 years after the first sign is noticed, and that's usually not necessarily because of not necessarily because the signs are not as present, but it's generally because of a lack of knowledge by physicians. So we wanted to look more at the early signs of PXE to really see what they are. So we completed 2 progression studies, basically. They were 2 separate questionnaires. The first one was conducted between November of 1998 and November of 2000, and it involved a detailed questionnaire being mailed out to all the individuals that were affected by PXE that had registered with PXE International, and that included 45 children and adolescents that were under the age of 18. The second study was conducted electronically, and it was completed between June 2006 and May of 2007, and individuals were emailed an invitation to complete an online questionnaire. 60 adolescents were invited, including 14 of the individuals who had completed the initial questionnaire in 2000. From these two studies, we basically had two study groups, and we wanted to accomplish two things. First, we wanted to simply describe the initial presentation of PXE in children, and we were able to do this by looking at the largest group of individuals, which was called the full cohort, and that was made up of 53 people who had completed at least one of the two questionnaires. Secondly, we wanted to study the progression of the signs and symptoms over time between the two questionnaires, and we did that by comparing the answers of 14 individuals who completed both questionnaires. So what did we learn from these studies? Looking at the largest group, which was the full cohort of 53 individuals, we found that 93% reported changes in their skin before the age of 18. And the average age of onset was 14 years for males and 8 years for females. Now, it's important to note that these ages of onset are generally younger than have been reported in the past, but the other cohorts that were reported on were also very small. And there were a few males in this group that were diagnosed earlier than usual, and that can kind of skew the ages, so we don't know if that's statistically correct or not. So that's just something to remember about the age of onsets for this study, is that this is a small group and that it's possible that there is some skewing towards younger years here. Other initial signs were 4% reported changes in their vision, and then 3% reported a diagnosis of a sibling as their initial sign of PXE. So overall, the diagnosis methods that were seen was 51% reported that skin biopsy was the method most used for diagnosis, and then the rest was generally a combination of skin and eye examinations with biopsy or something similar. This really didn't surprise us, generally because at the time and still today, skin biopsy is the usual method of diagnosis. Since the genetic test didn't come out until recently, this is still the most prominent diagnosis method used. Looking further at the vision changes that were noticed, the early changes reported included the peau d'orange and the angioid streaks, and the average age of diagnosis was 12 years for females and 14 years for males for the peau d'orange discoloration, and then the average age of diagnosis was 14 years for females and 15 years for males for the angioid streaks. In addition to looking at the skin and the vision changes, the other signs that we studied included cardiovascular and neurological changes, as well as gastrointestinal signs. And the significant changes that we found were that 14% of participants reported weak pulses, and 10% of participants reported experiencing medication. And while these signs were significant in our study, it's important to remember that this, again, is a study with a limited number of participants, and further studies are seriously needed to confirm this data before we know if these types of signs are really significant. So looking further into the progression of PXE in the early stages, and this is looking at the 14 individuals who completed both questionnaires. So again, this is even smaller than the large full cohort of 53 individuals. We found that 14% of individuals reported progressing from normal appearing skin to bumps or papules, which is here listed as a 1 under this picture. This is a scale that we use to measure the progression of PXE. And then 30% reported progressing from normal appearing skin to plaques or coalesced papules on the back of their neck. And this is a progression over the 7 years between the studies. And no one reported progressing from normal appearing skin to lax or folded skin during the 7 years between the studies. As far as progression of eye signs, 50% of individuals reported developing peau d'orange or angioid streaks between the time of the first questionnaire in 2000 and the second questionnaire in 2007. We also completed a quality of life analysis, and that was basically asking participants about how PXE affected their life over a period of a week and then over a period of 3 months. And we found that 17 to 21% reported feeling embarrassed about the changes to their skin, but 50 to 70% reported that PXE did not affect their leisure activities such as sports or reading or going to school or anything like that. As far as the effect of vision changes, 14% of individuals reported wearing protective eyewear when they were playing sports, but again, this is a small study set, and we would need to do further research to confirm this data. So as far as the study goes, the conclusions that we came to were that at 12 years of age or younger, approximately 29% of study participants reported the appearance of plaques in the neck area. And on top of that, 21% of participants reported having plaques in the back of their neck by 13 years. So that just confirms our initial understanding that the skin changes are the earliest signs of PXE and that they can progress over a period of 7 years. And we also found that approximately 50% of participants experienced retinal changes by the age of 21. Only one participant reported any type of bleeding in their retina before the age of 21, and that's 1 out of 53 participants. So that's a very small number. But again, the participants did report angioid streaks and pudor-orange discoloration. So further analysis is definitely needed to compare the progression of PXE in pediatric individuals based on this study. This was really just an initial study looking at the largest group of pediatric individuals affected by PXE that we've seen. So that was a really short just overview of the study that we conducted and the results that we found. And now I'm going to open the floor from Chris to see if anyone has any kind of questions.
Again, I would remind you that you can type your questions into the question box on the right side of your monitor. I think a few people weren't here at the beginning. So if you have any questions for Emilie about this presentation, I had a few other questions that aren't related to that. So if you have any questions, you can type them in now. Emilie, thank you very much. While we're waiting for some questions, I have a few. You can see our future webinars on this slide. The next one will be September 23rd about mice and PXE. And all our past webinars will be up on our website in audio and PowerPoint. Okay, Emilie, do you have any data on how adult responses to the age of onset question correlate with reported age of onset by children?
The most recent study that was done showed that the average age of onset in adults who had been diagnosed was 14 years 14 years for the age of onset, and then an actual diagnosis about 10 years later. But recently we've determined that it's possible that the progression of PXE is different in individuals who are diagnosed earlier just because it's being followed, as opposed to individuals who are diagnosed later, and it's possible that because you're then looking back at the progression of the earliest signs of their PXE, that there's some kind of biased or skewing. So again, these results were in a small study set, so it's possible that the small sample size might have skewed the onset, but generally what we've seen is a later age of onset in adults because those groups that have been studied are much larger. So for example, PXE International is in the process of reviewing, actually it was part of the 2000 questionnaire that I spoke of at the beginning. They are in the process of reviewing the data on the adults who completed that study, and the results of that study have shown a much later age of diagnosis. So I think it's around 15 or 16 years in that particular study.
Thank you. We have a couple questions about the study. Are the percents given, all the percentages you gave, are they related to the 53 individual samples? Are they related to the 53 individuals?
The early ones I gave, so the ones that were describing the initial signs, so these percentages here, the 93% that reported changes in their skin, those were all done on the full cohort where we were just trying to describe the initial signs of PXE. So yes, those are the 53 individuals. The percentages regarding the progression of PXE, so the 14% that progressed from bumps or papules and the 30% that progressed to coalesced plaques, these percentages are for the 14 individuals who completed both, because we couldn't do a progression study if we only had one questionnaire completed, which was for the 53 individuals. So the 14 individuals, we had an answer in 2000 on how their skin appeared, and we had an answer in 2007 on how their skin appeared. So we were able to do the progression of the skin changes for those 14 individuals. So these percentages are only for the 14 individuals.
Okay, thank you. Another question, is it fair to assume that further follow-up studies of pediatric to young adulthood are needed?
Absolutely. PXE International is actually in the process of looking at this because the diagnosis of PXE is generally delayed by 10 to 12 years. It's usually very difficult to find pediatric individuals with PXE that we can ask to participate in our studies, which is why this study was one of the first to involve 53 individuals at all. But we are definitely in the process of trying to look further at the progression of PXE and to try and find a large enough study group so that we can really be sure about the results that we're getting. This was just an initial look.
Mm-hmm. And a follow-up is how can you suggest how best to find appropriate participants? And how could you exercise good control of data gathering methods and follow-up?
Well, currently we're working with groups, and PXE International is attempting to expand the registry to younger individuals affected by PXE through a lot of networking sites and things like that. So we're really trying to get knowledge of PXE out there more so that it can be diagnosed earlier and we are able to find individuals to participate in early studies. We are working with more online technology to complete questionnaires online and be able to track data electronically over time. The initial questionnaire was one that was mailed out, and so now PXE International has an online questionnaire that you can fill out, and what we've done is we're in the process of completing the questionnaire at one point in time and then doing a 10-year follow-up so that we have the electronic data to track over the 10 years. So that's going to help more in the data tracking and being able to follow the progression of PXE for some individuals from the beginning all the way till their mid-years and hopefully until they're late in life so that we can really understand the progression of PXE.
So there's another follow-up question to that is can the PXE registry as it exists now be used as a participant source? And I think that I think you've answered that in the sense that people can now do the questionnaire longitudinally over time, and we can track it electronically.
Absolutely, absolutely.
Okay, we have another question from an adult who said he was diagnosed after he had eye problems when he was 35 and he had no apparent skin problems until they really looked closely. So he's asking, with regard to the children in this study, what was the most common precipitating symptom that resulted in diagnosis?
Again, that was the skin changes. As we saw, 93% of individuals had already experienced skin changes before the age of 18, and the skin biopsy was clearly the most used method of diagnosis. So skin signs are definitely the most, I guess you would say, noticeable factor in early diagnosis of PXE, and I think that, again, that shows a lack of knowledge for the physicians who diagnose PXE, because of that also reinforces the delay in diagnosis. So if this individual didn't have skin changes, then it's likely his diagnosis or her diagnosis was even more delayed because of the lack of outward appearance of the skin changes caused by PXE. So we did see that skin was the earliest indicator, but again, it's possible that this is a skew to our data because these are individuals who were diagnosed early because of their skin changes.
And can you go over again the data that this study gave about the eye manifestations on these children?
Sure.
This is another question from the same person.
So again, looking at the total cohort of 53 individuals, let me see. The vision changes, the pewter orange was seen early. It was seen 12 years for females, and the average age of diagnosis was 12 years for females and 14 years for male. And the angioid streaks, the average diagnosis was 14 years for females and 15 years for males. And looking at the progression over the 7 years, by the age of 18, 50% of individuals in this group of 14 had already reported developing these types of skin changes.
Do you mean vision changes?
Right, vision changes, sorry.
Right, okay. Okay, another couple questions. Do you have enough data yet to look at the correlation between age and age of onset and severity?
We do not at the time. We have actually looked at genetic mutations in severity and have not been able to find any type of correlation between what we consider the genotype, which is the genetic mutations causing the PXE, and the phenotype, which are changes in vision, changes in skin, the severity of symptoms. And in the study that we published in 2008, there was no correlation found between that, between the genotype and the phenotype of over 300 individuals. So because there was no correlation found in that large group, we certainly were not able to find any kind of correlation in this smaller group of individuals.
Yes, and that's pretty typical. We have not seen it's very unpredictable, the severity of PXE. And the second part of the question is, in the 14 individuals, did their progression, was it roughly the same, or did some progress faster than others?
Well, because we only had the two answers from 2000 and then 2007, we weren't able to look at a rate of progression. So if it progressed, very quickly in the early years between, so like if it progressed very quickly between 2000 and 2002 for one individual, and then progressed on a more gradual basis between 2000 and 2007, because we didn't have any points in between, we weren't able to determine any type of rate of progression. It was more, did you have angioid streaks in 2000? Did you have angioid streaks in 2007? It was more of a yes-no question, not so much of a progression. So we weren't able to determine a rate of progression for any of the individuals.
Okay. And then another question, did any of the individuals in this study report retinal bleeding?
One individual reported retinal bleeding at the age of 18, but again, that's one individual out of 53 individuals.
Okay. Okay, right now I don't have any more questions. If anyone has another question, you can type it into the question box, but for now, I think we are finished. And if you would put that final slide on, Emily. There were a couple questions that were more for our medical director, so I've responded that I will write the answers to those questions later. They don't have to do with this study. And you can always send questions to info@pxe.org, or you can call the office at the number given on the screen. Again, we have our next webinar is September 23rd, and it's called Do Mice Have PXE and Why Study Them, and that's Dr. Yoni Wido who is presenting. You can register from our website, and there is the URL. And all of our webinars are recorded, and you can see the visual and listen to the audio on our website. They're not up there yet, but they will be soon. And Emily, I'd like to thank you very much for presenting the results of your study. You did this as part of your master's program, and you've done a very good job, and you have some congratulations from some of the attendees today, too. So thank you for being with us.
Thank you very much.
All right, with that, I think we are, so I have a few more questions. Did a male or a female have the retinal bleed?
I'm not sure. I don't remember exactly if it was a male or a female or not. I do remember that they reported the bleed very late. I think it was when they were 18, or they might have actually been 20 or so when they had the bleed, but I don't remember their gender.
Okay.
Sorry.
All right. Well, that's it. Thank you very much, everyone, for being part of the webinar, and hope to see you next time at our next webinar. Thank you again, Emily.
Thank you.